Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. This is another call for the virus book we're going to be putting out. And today I have Seyedtaghi Takyar. He goes by Shervin. He's at Yale University. He has a PhD in microbiology and molecular biology from the University of Queensland, Australia. And he's worked on projects that include viral vectors, and he's got a lot of interests. So, Shervin, thanks for coming.
Sure. Thank you for inviting me. Get attention to this subject.
Yeah.
Very timely.
Yeah. So, I wanted to get people from diverse backgrounds to answer questions. So that's why I have you here. Yeah. So let's jump into the question. So is there any form of life you know of that doesn't have viruses associated with it?
Well, based on what I have read and learned, no, it seems like viruses are pretty ubiquitous. They're present in every form that we have found. It seems
okay.
Like.
And then based on, your observations, do you see them as alive or contingently alive when they're inside of a cell, or what are your thoughts there?
Yeah, so this is a very interesting question. I think a lot of people have been talked about this, thought about it, philosophized about it. I think so. Maybe it's like saying, do you think an algorithm is a computer or not? An algorithm can do functions, but inside a computer. So viruses are programs. In a sense, I can say they're alive because they do replicate, they do program for functions, but of course they don't have all the properties of an organism. You cannot say a virus breathes when it's in its viral form, doesn't metabolize. It has a program for metabolism, but it doesn't metabolize per se when it's in this virus form, in its virion form, or at least not as much as what we are used to. However, has a program for it. So if we call it a life form, we can say one of the most advanced forms when life has become a program, an algorithm. If we absolutely want to have a distinction as a rigid line, which I believe nature doesn't have. Nature doesn't have these kind of very strict lines. But if we were to draw a line like that, I would say viruses are at least footprints of life, right? So it is something that cannot be formed by anything but a live organism. When you find a virus, they have order in structure, they have a sequence of functions, their program has an aim. So that's why I think it's more, I think a lot of questions are like that. It's more interesting to understand their place in life than calling them alive or not. They definitely have the potential for life. They have very high potential. They have a program. They have a plan for life, but they don't have all the specificities of a live organism. So,
gotcha. Okay. In the context in which you understand them, why do you think there are different latency periods when someone first is infected, or some creature's infected? Some may be hours, days, some may be years. Why do you think there's this variable period in which someone first gets exposed to a virus and the time that it turns pathogenic and affects them negatively?
Yeah, so I think, again, in this respect, they also follow rules of life, rules of nature, really. It's all about fitting, right? When does a function that you do fits in your milieu, in your environment? I mean, we see examples of that in our everyday life. As an example, you can get one of the best scientists in Europe or US and put them in a desert, and they won't be able to function. They fit in a certain lab, in a certain country, in a certain town, in a certain university. So a lot of functions in life are about fitting. And when can you express yourself? And how can you express yourself? I think viruses abide by the same rule. The fact that So there's this whole time, like there's a time with any virus that they can express themselves, they can replicate more, and it's time for them to come alive, quote unquote. That time is dictated by their environment, which is within the cell. Now we are talking about an integrated virus, the virus inside the cell. Viruses life cycle outside also, we know that. But that's more of a kind of a diluted,
Well, do they have a life cycle? I thought they're just virions that passively float around outside cells. But do they have a life cycle even then?
Let me tell you, I have a more of an all-inclusive view. I call all of it life. There are parts of life. They're more in a more asleep state. They don't. Yes, the program is there in the virion and it's ready to bind and then express a function. Inside the cell is the same. Inside the body is the same. They may not express parts of their genome and just be in that semi-live state until the time comes. And that time inside the cell is dictated by a lot of factors, that aging that the cell is going through. Right now, we are talking about increased mortality from SARS-CoV-2 in patients who are older. It's something we see. Patients who have certain types of disease. A certain type of disease or aging is a code for the virus. Here is the time, meaning certain things, certain parts of this whole process that you need to grow are now available. That's the code. To us is aged patient with a heart failure. To us is a heart failure. To a virus inside is a code for certain prerequisites are available now. So the period that the viruses go in and they do not express all their genome also follows the same rule. They need certain prerequisites.
What form do you think these codes or prerequisites take? I mean, like bacteria can do quorum sensing and have contingent behavior based on that. Maybe viruses are able to use cellular machinery to do their own form of quorum sensing. How many infected cells are nearby? What's the density? Okay, there's enough. Now we go into lytic action.
Yes. So now this is of course not my area of research. I am like you, reading and trying to understand some very, very interesting information had recently come out, particularly in phages and how they have a social life. They talk to each other. And yes, it seems like in most of the functions that viruses do, including what we call quorum sensing, which is, as you know, is a term that came from bacteria and use it. They do message each other. And these messages are translated through the cell's translation machinery. Now, this can be bacterial machinery. We're talking about phages, right? So yes, they use the machine, they use the factory per se inside the bacteria or inside the cell to translate their messages and then talk to each other. They do that. But this is very recent. It is very interesting. It's just opening up a new field. But based on some very good research, it is recent. Dr. Sorek has published that, and there was also a news feature in I think it was Nature that talked about this. It's fascinating. It's just the social behavior, and in the middle is the.
So where does this in phage? Where does this behavior happens inside of a cell, like just within a cell, or across cells?
Yeah. So what they were talking about is inside bacteria. So they almost have to, right? With a condition that I said they are using the translation machinery inside the bacteria to translate their message. So they basically have to be inside, use that machine, create the messages, and then talk to each other. Some of these messages are question and response, right? Does a bacteria grow? How many bacteria are around? How many of these phages are inside the bacteria? It's a pretty complicated scheme that is coming out of this.
Yeah, that leads into a question I was going to ask you. So phages communicating through bacteria, I guess they do it through plasmids. Because I was wondering, in human cells, I wonder if viruses are able to have the cells put out specialized extracellular vesicles and use that as a form of communication as well.
Yes. So this whole science of extracellular vesicles is becoming very interesting. I think in one of the conversations we talked about, we touched on this subject a little bit. And so there have been good research showing that this kind of communication, meaning a particle containing genetic material being produced in one cell and then transferred to another cell, is being utilized in some form, for instance, in cancer.
Like cancer cells put out different ones because they're cancerous, but I wonder, like, again, phages are probably getting bacteria to put out specialized plasmids. And I'm thinking that, viruses are causing human or mammalian cells to send out specialized extracellular vesicles, as communication.
So I can say that's entirely possible. The specific examples of that, viruses causing that, it's very likely. I cannot remember. And by the way, the cancer, for instance, one of the most famous types of work that was done was cancer cell modulating the environment distally, like away from the cancer cell, modulating the environment by regulating some of the supporting cells. That was the fascinating part. Not just that the vesicles are out, no, just that the existence of these vesicles causes non-cancerous cells to become more responsive to a metastasis. That was the fascinating part as a function.
I think they call it niche construction.
Yes, a niche. So they are creating a niche. That word, of course, came from stem cell work, but now we use it as tumor niche. It's one of my one part of my research is about that, and endothelium, how the role of endothelium, the vascular cells, in creating that niche. And if the question is, for instance, are the virus is it possible for viruses to create a niche, let's say for cancer to grow? Yes, I believe that it is. How much data I have, I do not have solid data yet. I'm looking at some footprint. Is speculation at this point. We are looking at things that viruses can do, and through that increase the potential for tumor growth, which is equivalent to creating a niche for a certain cell to grow.
Okay.
Meaning that we have similar functions.
How far do you think the cooption of cells can go by viruses? I guess, I'd have to provide a specific example, but again, if they can do quorum sensing using the cell's machinery, I wonder if they're doing some immune system monitoring from within the cell. I guess they must be, because this was a question I was going to ask you now, is some viruses will be, pathogenic, but then they'll go into a latent period, like, hepatitis for years or decades. And then something happens in the host. Maybe they're immunosuppressed or they're senescent, whatever it is, and then they turn lytic and like rats leaving the ship, and they blow everything open and then destroy, What do you think governs that behavior, and how is that monitoring accomplished to do that?
Some of that, as I said, is basically the fit. For instance, hepatitis C, I worked on hepatitis C years ago, and hepatitis C gets reactivated. Hepatitis C is one of the wonders of virology. It's a simple virus, actually, but can be latent for a long time, and you may not even have difficulty finding it. We were one of the groups who made a very highly sensitive assay for detecting hepatitis C virus. And then something happens, and it's an injury, it's a trigger of some sort, and the viruses start to replicate. Now, at that time, a lot of it seems to be related to the immunity coming down. So immunity is the other side of this, has been programmed to look at anything foreign, anything that doesn't belong, and taking them out, and taking the cells that harbor that. So now, as soon as those go away, or a mimicry happens, meaning the virus now, or the code that is putting out, mimics one of the codes that our immune system is used to, meaning is recognizing them as self. If that happens, then viruses can grow. We usually use viral mimicry in the other sense, how viruses mimic some antigen and in cellular sense, and then do things. So this is the other way. There may be induced tolerance, and the viruses can then grow. So that is how, at least some of the events in the interaction between viruses and cells they lay dormant. As I said, a prerequisite. I put it in the general category of prerequisite. It can be that immune system has to be suppressed for them to be able to grow and have their antigens out or expressed, and then they replicate further.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
But how far this cooption can go, that was your more general philosophical question. I can tell you, I think, as far as any function, any biological function goes. I mean, we are talking about genomic elements, right? We are talking about genomic elements that may comprise up to 50% of our genome.
Oh, I thought it was like 8 or 10%.
So 8 or 10% are endogenous retroviral elements. So those are the ones that mimic very closely a retrovirus. But then we have all these other groups. They're called lines or signs, interspersed elements. These have, they are very similar. They have similar functions to endogenous retroviruses. So some people believe that those are retroviruses that have lost part of their genome, but they retain some of the functions. And if we get into the lines and signs and the group, they do a lot. They can form and regulate how a cell is, what a cell is, what a cell does, which gene comes up, which gene is silenced. They do very, very basic elements, the functions.
Okay.
So that is a part that I'm interested in because, as you know, I work on microRNAs, and these elements, these interspersed elements, they can program the microRNA function on some mRNAs, which I think is fascinating. They're basically sculpting the expression of the genome at multiple levels, saying, well, here's a gene that has to be silenced now. They, using your word, co-opt a microRNA to now come and sit on this particular gene, this RNA, this mRNA, and to go away, to get degraded. They co-opt transcription factors to come and sit on a particular gene for that to be expressed. They actually function on chromatin modeling. Chromatin modeling, you probably have heard, is basically the basic way eukaryotic cell will select which genes to express and which genes to keep away from inhibition. So we are there. We are basically every step of gene expression, every step of life. They're having a role in.
Yeah, amazing. Do you think viruses at all are like a swarm? You know, like if you have bees, you have drones and workers and the queen, et cetera. They're all similar, not exact phenotype, but genetically they're all, I don't know how similar, but probably incredibly similar. But they're all bee. They all work together. They're like a swarm. And in viruses, I've read about quasi-species. So these would be viruses that have slightly different sequences, but they're all pretty much morphologically and functionally the same virus. So do you think that when an infection happens, it's just a pure, one sequence virus, or do you think that there's a swarm of quasi-species that all have maybe slightly different ability, and that's what does the infection?
Okay, so this is a very interesting question you bring up because it has some philosophical implications about evolution, social evolution in general. Now, quasi-species, let's just talk about quasi-species for a second. Quasi-species are different sequences of the same virus in a host, right? So quasi-species, like I worked with that when I was working on hepatitis C. We sort of know how they're generated. There are polymerases that viruses use and all other organisms use to replicate their genome. Now, these polymerases can be permissive, meaning they're not high fidelity, meaning they allow some mistakes in sequence. That's how quasi-species are generated. The polymerase of hepatitis C is permissive. So there are quasi-species. There are many sequences with some mistakes, with some variation that come out in every host. Now, why is this important? Quasi-species is one of the ways that they may work the same way that mutation works in evolution. There are types that may be more resistant to or more capable of survival, right? So in that sense, quasi-species can be a tool for survival. You have sequences, for some reason, one of them may be better fit, more fit for survival. Now, are these cooperating? Are these, like quasi-species in particular, are they cooperating? Well, it goes back to how much you submit to the whole theory of, for instance, selfish gene, that philosophy, that here is a particular sequence, a particular life form, and it replicates more of itself, and all the rest is manifestations of that. You probably have heard about the theories of Dr. Wilson about ants and bees, and how different ants or bees are extensions of the genome of the queen. And each of them have an expression of their own gene, but they're extensions of the queen. So in that sense, they are all working together to propagate the genome of that queen, of that queen bee or queen ant.
Oh, quick question here. I don't know if you know. If not, it's okay. But are the different types of bees just epigenetic variants with different markings of the others, of the queen, or they literally have different sequences or additional sequences or chromosomes?
So there are epigenetic differences in each bee. The genome, the content of genome is coming from the queen. There are different kind of combinations between the queen and the father, and it comes out in different bees, but the content is coming from them, from the two parents.
No, I know that, but what makes, on a genetic level, what differentiates a worker bee from a drone from a queen? Is it literally they have different sequence of base pairs, or is it just at the level of epigenetic marks that's the difference?
Different expression. It's different expression, not this sequence. It's mostly different expression, just meaning the epigenetic.
Okay.
So in that sense, viruses can also do that. They have quasi-species. There are different sequences, maybe with different chances of survival. The more variable they are, the bigger chance they have for surviving one of them, which is coming from an initial copy, right? Initial sequence that has variations. Now, the cooperation, if you mean by that different quasi-species doing different things as a team, the way that ants do or bees do, I don't believe that. I have not heard of that. That's a higher level of cooperation or eusociality, if you've heard about that. And classically, you say only humans and ants or bees, wasps, maybe some of them do that, meaning different members of a society doing different jobs and then getting together in a big hierarchy. I have not heard of viruses doing that.
I think of a one virus, one cell infection model. But so there's a couple questions wrapped up in this. Are there any circumstances where it takes multiple viruses to infect a cell successfully? What if it takes 2 or 3 to fuse to a cell membrane? Is there any papers on that, any observation of that? And then once a cell is infected, can it be infected, let's say, by, multiple different quasi-species of the same virus? And because it's infected that way with multiple, again, variants of the same virus, perhaps that infection is more successful if that sort of thing happens.
I have not heard of research that can answer each of these questions completely, but there are some bits of information that may fit to parts of your question. For instance, we know that there are times that we need to have a certain number of viruses per cell to have a productive infection. Now, is that a stochastic event? Is it just that because all the viral infections start from binding, as you know. There's some binding event that should happen. And binding events have this kind of this stochastic nature to them. A successful binding, per se, may need a certain hit, a certain number of hits for one of them to be successful. Now, if that translates into actually 10 of them have to bind and then do something to the cell for one of them to get in, I don't know. People may have been looking at. The recent social life of viruses, which was talked about, was in phages, goes a step further. Different phages talking to each other about how many we are, what should we do, where are we going. There is some information there, some finding there, and that is much more advanced than what we know about other viruses. A third way of answering your question is with the clinical observation. So I'm coming out of the cells, I'm going back to my medicine part.
Okay.
We know that the progression, the viral progression for hepatitis viruses varies in the presence of HIV. If you have hepatitis B plus C, if you have hepatitis P plus delta plus C, the progression changes and becomes more fatal. So there are these bits and pieces. It seems like they can cooperate at times, like in the case of HIV and hepatitis B and hepatitis C. If we call that cooperation, the infection becomes more severe, harder to treat. So that we also know. Now, what exactly is the mechanism? I don't know about that. Maybe some people have found things. I have not heard it.
Okay. Yeah, if I was able to make an isolate of a virus where it's just one sequence, that's it, and I would, then I was to infect an animal, and then I was to infect another animal with the normal wild type, where you get all these quasi-species and you get like this whole, again, swarm of variation. Do you think that the infection would be a lot more effective if I used a varied population versus like an isolate of one sequence only?
Yeah, possibly. The chances of survival are high. Yes, I would put my money on yes, because you will have higher chance of hiding from immunity just because you have variation within your quasi-species. If you have a virus that retains its replicative ability, has the replication machinery, has a variation in sequence that can hide from the immune system better, that one will win. We are talking about the same thing. I was talking to a patient of mine 2 days ago about this whole issue. I talked to this patient about COVID-19 and where it came from. And this patient of mine believed that this is like flu, and flu is new and every year, and it can be very similar. And I said, well, it is correct to some degree. It is the mechanisms of viral hybridization or variation happens in both flu and SARS in principle. The difference is that with this SARS-CoV-2, if the variety of sequences that you're generating, if within them you have a variation, a type that has a sequence for the code protein, for instance, that immune system has not seen, cannot mount an immune response against, that sequence will have higher chance of survival. However, the experiment you were suggesting, that if you get one sequence and you put it in versus the variety of sequences, I hesitated a little bit because, as I said, making variation in sequence is an inherent property of the polymerase of some viruses, meaning that even if you get one sequence of hepatitis C and put it in an animal or in a cell, since the polymerase is promiscuous, it allows mistakes. Very soon you will have quasi-species anyway. Now, putting in quasi, that increase the viability of the virus. I think it still does. It will still increase the chance, but we would never know until we test it.
Yeah, what if we revisit viral titer and look at it not just as the amount of virus, but look at the variation in the viral load that you're getting? If we include it that way, maybe titer takes on a different or an additional meaning now, or an additional nuance.
Yeah, some of that has been done, like the variation, like the quasi-species to some degree has been done. I don't know if there has been a clinical association. There might have been in hepatitis C literature. There might have been because there was some talk about quasi-species and how extensive it is and viral progression. But I'm not entirely sure about that. I can check that and get back to you.
Okay. Yeah, I was just wondering. You study a lot of RNA and everything. So from what I understand, there is an epigenetics to RNA as well as DNA. Do you think that viruses acquire epigenetic marks when they're inside a cell? And do you think that leads to further variation? And can quasi-species be defined not only as, variants with different sequences, but variants that have different epigenetic marks?
So I think we are talking about different parts of the expression, right? Like, for instance, RNA can be spliced in different forms. The same DNA element can make RNA, and then those RNAs can be spliced to different variants of the same gene. And this is one of the things that, as I said, lines and signs can affect. They can affect splicing. These are all, the sequences are coming from the same gene, the same DNA. When we say epigenetic as DNA part and the RNA part, it means that there are different mechanisms that affect the DNA or affected at the RNA stage or the DNA stage. Epigenetic is just a very general term for many, many different processes that can happen. So now viruses can definitely get inside the cell and affect the epigenetics of the cell. One of the ways is what I just told you. microRNA mediated regulation is one of the epigenetics forms of regulation. And viral elements can do that, can affect the microRNAs within the cell. So yes, they can affect the epigenetics of the cell. Now, that's another step, because you're asking about that, right? You're asking about epigenetics of the virus, right?
I'm wondering, do viruses acquire epigenetic marks? Yeah.
I'm not sure. They might. I mean, the machinery, the possibility is there. Can we call it an epigenetic mark for the virus? The viruses are more fit. I mean, they can become of the epigenetic mechanisms. I think it is possible. But again, this is a specific instance. If somebody has found, I cannot remember. I'm just trying to think. For instance, a lot of people talk about chromatin remodeling for epigenetics. So I'm not sure about the answer to that question.
Yeah, because if viruses can acquire epigenetic marks, that would be another source of variation. So now I could have, if I'm infected by a virus, I may be infected by many variants in the nucleic acids that comprise the virus. And each of those variants may have a different set of epigenetic marks from, past infection.
So I'm trying to go back and think about mechanisms of epigenetic variation. So one of the most famous one, chromatin remodeling. Chromatin remodeling is which gene is expressed and which gene is not. So if we talk about a simple virus, like hepatitis C virus, right? Hepatitis C virus, well, I mean, we're out of it. It's an RNA virus, so we're out of chromatin. We're not even talking about chromatin. We're not talking about methylation either. MicroRNA is the one that I'm hesitating about. If, for instance, a quasi-species are sculpted or formed that can co-opt a microRNA better than others, for instance, it is possible. I mean, we are putting a sequence in a soup, in a conglomerate of sequences within the cell. And as we talked about, there is this whole endogenous RNA circuit. RNAs talk to each other. So now, if within the evolution of a virus, if during the evolution of a virus inside the cell, some sequences are selected based on interaction with other RNA elements, for instance, is that a possibility? And then that RNA element, that RNA type, that variant survives more. I think the possibility is there. It is possible that some viral sequences are more fit. There are parts of the viral genome that do not tolerate as much variability. For instance, the polymerase structure. Parts of the polymerase structure do not tolerate much variability. Any virus that loses that dies. Any sequence that loses that dies. But there are parts that tolerate a lot of variability. SARS-CoV-2, we're talking about flu, right? And the coat protein for flu or SARS-CoV can vary, can be the product of a hybridization. Now, if there is an intracellular counterpart to that, it is possible that there are certain sequences that are fit. I don't know of a specific example.
Okay.
But possibility is there for that type of epigenetic.
Yeah, I wonder if methylation could happen, et cetera. But I wonder how much, if any, epigenetic variation happens to viruses. So
yeah, so that would be the herpes viruses. Those are even more sophisticated viruses that have DNA, and then they go through sequences. Methylation of viral sequences there are some virologists who work on that. It's a very interesting area. I don't think I know enough about that.
Okay.
I try to stick with what I know a little bit more about.
Yeah, no problem.
For these viruses, for the RNA viruses.
I'm trying to give you really softball questions, just kidding. But so
yes.
if I get infected by a virus, let's say, I don't know, a bat flies out of a cave and bites my neck and infects me with a virus, and I call myself number one, and then I go home and I get my wife sick and she's number two, and she gets someone else sick, and the virus passages through 100 different people. How do you think it would change? And I know this is, depends on the virus, et cetera, but as a virus passages through many people, for instance, or many animals, how do you think they tend to change? Is there any direction they tend to go in? Less pathogenic, more latent,
It varies. It varies. That's all I can say. I mean, this whole again, the creation gives so much. This whole pandemic is because of that. It's because of passage of virus and going into one organism into another one and bringing sequences from each and putting them together. So viruses getting together and sharing sequences, for instance, to come up with a more fit virus. So it's an observation. This whole herd immunity, you probably have heard about it. The herd immunity is the opposite of this. So it really varies. It depends. It's a general comment or general question, and there are specific examples of it. I don't think the answer is positive or negative, yes or no. It's just different things at different times.
Okay. If there's a virus out there, let's say, again, I'll just pick some random one. So if there's a virus out there that makes people sick, and how would it be different if I got sick from someone that was like on death's door? They were horribly sick with a given virus. What do you think would happen to me versus if I get sick from someone that's like barely feels anything? They have the same virus, but they just, for some reason, they barely feel anything. How do you think that it would affect me differently if I was to get sick from one of those two individuals?
There is a simple answer to this, and that is if the person who is sicker, because of their lowered immune status or the higher replication of the virus, they may have more viral load to give you.
Well, if we assume viral load was the same, what would be another reason that it would be different?
That's a difficult one. That's a difficult one. Is it possible that you get it from a person who, because of the fitness of the virus, is sicker? That is possible. The example of that one about that's a quasi species with hepatitis C that I said I can look up. That's the example I know. Those are variations within one viral sequence, and I vaguely remember that there was some association with severity, but I'm not a hepatologist. I don't work on hepatitis C right now. I can check it and get back to you.
Yeah, I don't want to give you all this homework and stuff. I wanted your speculation, but it's okay if you're willing. That's great, but it's okay if you don't know the answer, because I don't think anyone knows. But your thought process on it is what is interesting, and that's why I'm asking. So
sure. I think it's possible. I think it's possible.
Yeah. Just a couple more questions. I know we're getting to the end. Do you think that so an infection, I'm imagining it as variants of a given virus, nucleotide variants, and also they have different epigenetic marks. So if I'm infected by a viral swarm of a given virus, do you think that the variants and the various epigenetic marks, which came as a memory from past infections, could act as an immune system for the viral swarm? Do you think that this could be its own version of an immune system? Because these variants and these different types of viruses may help protect it or allow it to adapt or become more fit to a given host because of past infections and past learnings and retention of these variants.
I think it functions in a way that evolution functions, right? Viruses survive if they are fit. Viruses are annihilated if they're not fit or if they fail in the competition with another virus. Those instances are known. For instance, when coronaviruses, it is known that some coronaviruses come in animals and then suddenly they're wiped out, they go away. They're fatal, they're very successful in the beginning, but what happens is another type of the same virus propagates, uses the same receptor, species is gone. So if you mean memory in that general sense, yes, it works like that. The viruses that exist right now are the fittest ones for this time. Otherwise, they won't survive. There is the concept of memory in the immune system. I'm not sure if you were asking about that also Because immune system, yes, immune system also keeps the memory, and we know a little bit more about that, the memory cells.
Yeah, the 2-second version of the question is, do viral swarms have an immune system? And now I'm defining what I mean by that is, well, could a viral swarm have an immune system because these variants will be the fittest survivors from previous infections? So the whole collection of them together may serve as an immune system for the viral swarm because of its past experience, you know?
Yes. So the cooperation between viruses, a little bit in phage, a little bit is known about. How, and then is that formed through an evolutionary process? It seems like. So an evolutionary process have caused types of viruses like phages to exist now. And these types of viruses that exist seem to be able to cooperate together, the example being phages. So different parts of your question have been answered to some degree about some viruses. It's interesting that you're putting it in as immune system, because as soon as we talk about immune system, we are talking about cells doing the specific things. I think we are a little bit away from calling it an immune system. Saying this, I'm not an evolutionary biologist or virologist, so I may be behind on this. I just have knowledge about little bits and pieces here and there. What I know is this. Yes, viral sequences that exist and are active at the moment, they have to be the fittest. Can these viruses work together? It seems like, based on some data in phage, putting this together, can we call this an immune reaction? Maybe instead of system, I'll call it an immune reaction or a response, a survival response. Can this be a coordinated response in a general sense? Maybe a survival strategy. Let me call it that.
Okay, very good. Yeah, so well, I appreciate you coming on the podcast. I hope I didn't give you too hard a time, but you did great with the questions. And what's the best way for listeners to find out more about your work, by the way?
I publish papers. Since we had this discussion before, I'm thinking about writing more for a general audience. But I have so far, mostly my work is in a specialty journals, recently in cancer, asthma. Through Yale, we have a mechanism that we try to collaborate to some degree with the media, particularly if there's an important message. I collaborated a couple of times with our media group. As things come up, I'm going to try to help with them more. My time allows. The conversations that we had today, the type of conversation that just bringing science to media, to people who are enthusiastic, I'm happy to help out with.
No, that's great. Shervin, I really appreciate you coming on the podcast. Thank you.
Sure. No problem. Thank you for bringing up. These are fascinating concepts. These are really, to bring this to the population, to society, and thinking at this level about them is amazing. Thank you for continuing to do what you do and bring these things up.
Yeah, no problem.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.